So, here's something kind of wonderful to think about as you're lying there, getting comfortable, maybe pulling the covers up a little. You are doing the very thing we're going to talk about tonight. You're settling in, your body is starting to slow down, your breathing is shifting, and somewhere deep inside your brain, a cascade of chemical and electrical events is already underway. Events that scientists have spent centuries trying to understand, and honestly, we're still figuring out. Sleep. The thing you do every single night of your life. The thing every human who has ever lived has done. And yet, for most of history, nobody had any real idea what was actually happening during those hours of darkness and stillness. Tonight, we're going to walk through that story together. How we went from ancient guesses about sleep being a kind of mini-death, all the way to the astonishingly complex picture we have now of sleep stages, circadian rhythms, and the architecture of a single night's rest. And we're going to do it slowly, gently, because there's no rush. If you drift off somewhere along the way, that's perfect. That's literally the point. Let's start at the beginning. And I mean the real beginning. Not the beginning of sleep research, but the beginning of people wondering about sleep, because humans have always wondered about it. You can find references in some of the oldest texts we have. The ancient Egyptians wrote about sleep, the Mesopotamians wrote about sleep, and almost universally, early civilizations treated it as something mystical. Which makes sense, right? Think about what sleep looks like from the outside if you don't know anything about neuroscience. A person lies down, closes their eyes, and for hours they're just gone. They breathe, their heart beats, but they don't respond to you. They twitch sometimes. Their eyes move behind their lids. And then they wake up and tell you they were somewhere else entirely. Flying, falling, talking to dead relatives, being chased by animals. If you didn't know what was happening biologically, you'd probably conclude that the soul was leaving the body, and that's exactly what many ancient cultures believed. The Greeks had some interesting ideas. Aristotle, and this is around 350 BCE, wrote a whole treatise called On Sleep and Sleeplessness, which is one of the earliest systematic attempts to figure out what sleep actually is. Now, Aristotle got a lot of things wrong. He thought sleep was caused by warm vapors rising from digested food in the stomach up to the head, where they cooled and condensed, and that this condensation somehow caused unconsciousness. It's a beautifully wrong theory. But here's what's fascinating about it. He was trying to find a physical, mechanical explanation. He wasn't satisfied with just saying the gods did it or the soul wandered off. He wanted a mechanism. And that impulse, that desire to understand the machinery behind the experience, that's the thread we're going to follow all the way through tonight. For a very long time after Aristotle, though, not much progress happened. The Middle Ages came and went, and sleep remained largely in the domain of philosophy and theology, rather than science. People slept, of course. They slept a lot, actually. And here's a detail that I find really charming. There's strong historical evidence that before the Industrial Revolution, many people in Europe practiced what's called biphasic sleep, or segmented sleep. They'd go to bed when it got dark, sleep for about 4 hours, wake up for an hour or two in the middle of the night, and then sleep again until dawn. This wasn't insomnia. This was just how people slept. The historian Roger E. Kirch spent years digging through court records, diaries, medical texts, and literature from the pre-industrial era, and he found hundreds of references to first sleep and second sleep as completely normal concepts. People would wake up after their first sleep and pray or talk or visit neighbors or just lie quietly in the dark. And then they drift off again for their second sleep. Think about what that means for a second. The 8-hour unbroken block of sleep that we treat as the gold standard today, the thing you're supposed to get, the thing your doctor tells you about, the thing that sleep apps track, that might be a relatively modern invention. It might be an artifact of artificial lighting and industrial work schedules, rather than some deep biological imperative. Now, that doesn't mean consolidated sleep is bad for you. It clearly works fine for most people. But it does suggest that our relationship with sleep is more flexible, more culturally shaped than we usually assume. And that's a theme we'll keep coming back to, the tension between what's biological and what's cultural when it comes to how we sleep. All right, so let's move forward in time. The real scientific study of sleep, the kind where people actually start measuring things, doesn't really get going until the late 1800s and early 1900s. And the key technology that makes it possible is something you've probably heard of, the electroencephalogram, or EEG. The ability to measure the electrical activity of the brain through electrodes placed on the scalp. This is where the story gets really good. A German psychiatrist named Hans Berger is the person who makes this happen. Berger is a fascinating character. He was born in 1873 in Neuss, a small town in Bavaria, and he originally wanted to be an astronomer. But then something happened to him during his military service that changed his life. He was thrown from his horse during a training exercise and nearly crushed by a horse-drawn cannon. At that exact moment, and Berger was very specific about this, his sister, who was miles away, suddenly felt an overwhelming sense of dread and convinced their father to send him a telegram asking if he was all right. Berger became obsessed with the idea that the brain must emit some kind of energy, some kind of signal that could be detected and maybe even transmitted. He spent the rest of his career trying to find it. Now, he didn't find telepathy, but what he did find was arguably more important. In 1924, after years of painstaking work, Berger successfully recorded the first human electroencephalogram. He placed silver foil electrodes on the scalp of a patient. Actually, his first successful recordings were from patients who had skull defects from surgery, so the electrodes were closer to the brain. And he detected rhythmic electrical oscillations. He identified what he called alpha waves, these beautiful, regular oscillations at about 8 to 12 cycles per second that appeared when a person was relaxed with their eyes closed. And he noticed that when the person opened their eyes or started concentrating on something, the alpha waves disappeared and were replaced by faster, smaller, more irregular waves, which he called beta waves. Here's the thing about Berger. He published his findings starting in 1929, and for years, almost nobody believed him. The scientific establishment was deeply skeptical. The idea that you could detect the brain's electrical activity through the intact skull seemed implausible. Some people thought he was picking up muscle artifacts or electrical noise. Berger was also not a particularly charismatic or well-connected scientist. He was quiet, methodical, a bit of a loner. He kept working, kept publishing, kept refining his technique, but recognition was slow to come. It wasn't until 1934, when two prominent British physiologists, Edgar Adrian and B.H.C. Matthews at Cambridge, independently confirmed Berger's findings, that the scientific world started paying attention. Adrian, who had already won a Nobel Prize for his work on nerve function, essentially validated everything Berger had been saying. And once Adrian's name was attached to the work, suddenly everyone was interested. That's how science works sometimes. The same finding gets ignored or accepted depending on who's presenting it. It's not fair, but it's real. And here's the sad part of Berger's story. He never really got the recognition he deserved during his lifetime. He was forced to retire from his university position in 1938 under pressure from the Nazi regime. The politics of that era are their own dark chapter, and he fell into a deep depression. He died by suicide in 1941. The man who gave us the ability to see the sleeping brain at work, who opened the door to everything we now know about sleep stages and brain waves and the architecture of the night, never got to see what his invention would reveal. But the tool he created, that changed everything. Because once you can measure brain waves, you can watch what happens when someone falls asleep. And what happens is not at all what anyone expected. Before EEG, the assumption was pretty simple. You're either awake or you're asleep. Sleep was thought of as a uniform state, like turning off a light. The brain goes quiet, you're unconscious for a while, and then you wake up. One state, on or off. The EEG showed that this was completely wrong. Sleep isn't one thing, it's a journey. It's a sequence of distinct stages, each with its own characteristic brainwave pattern, each with its own physiology, and your brain cycles through these stages multiple times every single night in a pattern that's so regular, so predictable, that scientists can map it out on a chart called a hypnogram. This beautiful, staircase-like graph that shows your brain descending into deep sleep and then rising back up toward the surface, over and over, all night long. The first person to really start mapping this out was a researcher named Nathaniel Kleitman, and if Hans Berger is the father of the EEG, then Kleitman is arguably the father of modern sleep research. Kleitman was born in what's now Moldova in 1895, emigrated to the United States as a young man, and eventually ended up at the University of Chicago, where he spent decades, literally decades, studying sleep with a persistence that bordered on obsession. He was one of those scientists who just would not let go of a question. Kleitman did all sorts of extraordinary experiments. In 1938, he and a research assistant named Bruce Richardson spent 32 days living deep inside Mammoth Cave in Kentucky, one of the deepest cave systems in the world, to see what would happen to their sleep cycles when they were completely cut off from sunlight, temperature changes, and all external time cues. They wanted to know, does the body have its own internal clock, or do we just sleep when it's dark and wake when it's light? They tried to shift themselves to a 28-hour day, 19 hours awake, 9 hours asleep, to see if the body would adapt. The results were fascinating. Richardson, who was younger, adapted to the new schedule fairly well, but Kleitman, who was in his 40s, could not. His body kept drifting back toward a roughly 24-hour rhythm no matter what he did. His temperature still rose and fell on its own schedule. He'd lie in the dark of the cave, trying to sleep on the 28-hour plan, and his body simply refused to cooperate. This was some of the earliest evidence for what we now call the circadian rhythm. This internal biological clock that runs on approximately a 24-hour cycle and governs not just when you feel sleepy, but when your body temperature peaks and dips, when certain hormones are released, when your digestion is most active. The word circadian comes from the Latin circa, meaning about, and dies, meaning day, about a day. Because here's the interesting wrinkle. The internal clock doesn't run on exactly 24 hours. In most people, it's a little longer, maybe 24 hours and 10 or 15 minutes, which means that without external cues, without sunlight, without alarm clocks, without the rhythms of social life, your sleep schedule would slowly drift later and later, day after day. The reason it doesn't, the reason you stay roughly synchronized with the actual day-night cycle, is because your clock gets reset every day by external signals, the most powerful of which is light. But Kleitman's most important contribution was still ahead of him, and it came through a collaboration with a young graduate student who showed up at his lab in the early 1950s, a student named Eugene Oserinsky. And what Oserinsky noticed, almost by accident, while watching sleeping subjects in Kleitman's lab, was something that would change everything we thought we knew about what happens when you close your eyes at night. He noticed that their eyes were moving, not all the time, not during every phase of sleep, but at certain points during the night. The eyes of sleeping people would begin to dart back and forth beneath their closed eyelids, rapidly, almost frantically, as if they were watching something. And that was strange, because these people were deeply asleep. They weren't stirring, they weren't about to wake up. They were out, but their eyes were racing. Now, Oserinsky almost didn't follow up on it. He initially thought his equipment was broken. The electrodes he was using to track eye movements, a technique called electrooculography, were giving him these bursts of rapid activity that didn't seem to make sense. A sleeping person's eyes should be still, right? That was the assumption. Sleep was supposed to be this quiet, uniform state where the body powered down and the brain went dark. So when the instruments showed all this eye movement, the natural first thought was that something was wrong with the wiring. And Oserinsky actually spent time checking and rechecking the equipment, swapping out components, making sure the signals were real. It took a while before he accepted that no, the machines were fine. The eyes really were moving. Here's what made it even more interesting. When Oserinsky woke people up during these periods of rapid eye movement, they almost always reported that they had been dreaming. Vivid, narrative, emotionally intense dreams. The kind you remember when you wake up, at least for a few seconds before they start to dissolve. But when he woke people up during other stages of sleep, when the eyes were still, they usually said they hadn't been dreaming, or if they had, the dreams were vague, fragmentary, more like fleeting impressions than actual stories. So Oserinsky brought this to Kleitman, and to Kleitman's enormous credit, he didn't dismiss it. He could have. Oserinsky was a graduate student with a somewhat rocky academic history. He'd bounced around a bit before landing in Kleitman's lab, and he wasn't exactly the golden child of the department. But Kleitman recognized that this was significant, and together they designed more rigorous experiments to confirm what Oserinsky had stumbled onto. They published their findings in 1953 in a paper in the journal Science, and that paper is one of the landmark documents in the history of sleep research. It formally described what they called rapid eye movement sleep, REM sleep, and established that it was a distinct, recurring phase of the sleep cycle, fundamentally different from the other stages. And this is where the picture of sleep starts to get really rich and layered, because what the discovery of REM revealed is that sleep isn't just one thing. It's not a single state that you drop into and stay in until morning. It's a journey with distinct phases, and your brain cycles through those phases multiple times every night in a pattern that's remarkably consistent from person to person. Think of it like this. If you were to watch a graph of someone's brain activity over the course of a full night's sleep, you wouldn't see a flat line. You'd see this beautiful, rolling, repeating architecture, almost like a landscape with hills and valleys, and each cycle takes roughly 90 minutes to complete. Let's walk through what that looks like, because it's genuinely fascinating. When you first close your eyes and start to drift off, you enter what sleep researchers call stage 1 of non-REM sleep. This is the lightest stage. You're barely asleep, Your brain waves, which during wakefulness are fast and irregular, start to slow down. They become these gentle rolling waves called theta waves, and they're slower and more rhythmic than the alert, jittery beta waves of your waking brain. In stage 1, you might experience that strange sensation of falling, that hypnic jerk where your whole body suddenly twitches and you snap back to awareness for a moment. That's your brain in transition, caught between two worlds. Stage 1 usually lasts only a few minutes, and if someone woke you up during it, you might not even realize you'd been asleep. Then you slide into stage 2, which is where you spend the largest portion of your total sleep time, roughly half of it, actually. Stage 2 is still relatively light sleep, but your brain starts doing something really clever. It produces these brief bursts of rapid rhythmic activity called sleep spindles. They look like little bursts of oscillation on an EEG, lasting maybe half a second to a second each. And it also produces something called K-complexes, which are these large, sharp waveforms that seem to help protect your sleep from being disrupted by external noises. Think of them as your brain's way of saying, I heard that sound, but it's not important, so I'm going to stay asleep. Sleep spindles, by the way, are thought to play a really important role in memory consolidation, in taking the things you learned during the day and transferring them from short-term storage into long-term memory. We'll come back to that later, because the relationship between sleep and memory is one of the most exciting areas of modern neuroscience. After stage 2, you descend into stage 3, which is deep sleep, also called slow-wave sleep. And this is where things get really quiet and really powerful. Your brain waves slow way down into these large rolling delta waves, big, slow, synchronized pulses of electrical activity. This is the most restorative stage of sleep. This is when your body does the heavy lifting of physical repair, releasing growth hormone, repairing tissue, strengthening the immune system. If you've ever been sick and slept for 14 hours straight and woken up feeling noticeably better, a big part of that recovery happened during slow-wave sleep. It's also the hardest stage to wake up from. If someone shakes you awake during deep slow-wave sleep, you'll feel groggy, disoriented, almost drunk, a phenomenon called sleep inertia. Your brain is so deeply offline that it takes a while to boot back up. And then, after spending some time in deep sleep, something remarkable happens. Instead of staying in that deep, quiet state, your brain starts climbing back up through the lighter stages, and then it enters REM sleep. And REM is wild. Your brain activity during REM looks almost identical to when you're awake. The EEG lights up with fast, desynchronized waves that are strikingly similar to what you'd see in someone who's alert and engaged. Your eyes are darting around. Your heart rate becomes irregular. Your breathing gets faster. And yet, and here's what's really strange, your body is essentially paralyzed. During REM sleep, your brain actively inhibits your voluntary muscles through a mechanism called atonia. You cannot move. And the reason for this is probably obvious once you think about it. This is when you're dreaming most vividly. And if your muscles weren't shut down, you'd be acting out your dreams. You'd be thrashing around, running, reaching for things, maybe even walking. Which, incidentally, is exactly what happens in a condition called REM sleep behavior disorder, where that paralysis mechanism fails. People with this condition physically act out their dreams, sometimes violently, and it can be dangerous for them and for anyone sleeping next to them. It's a relatively rare condition, but it's a powerful reminder that the muscle atonia of REM isn't just a quirk, it's a critical safety feature. So that's one cycle. Light sleep, deeper sleep, deep sleep, back up, then REM. About 90 minutes, give or take. and then you do it again and again, 4 to 6 times per night, depending on how long you sleep. But here's the thing that makes it even more interesting. The composition of each cycle changes as the night goes on. In the first half of the night, the cycles are dominated by deep, slow-wave sleep. You spend a lot of time in those big, restorative delta waves. But as the night progresses, the slow-wave portions get shorter and the REM periods get longer. By the last cycle or two before you wake up, you're spending most of your time in REM, which is why your most vivid, most memorable dreams tend to happen in the early morning hours. If you set your alarm early and cut your sleep short, you're disproportionately cutting into your REM time, which means you're cutting into dream time, but more importantly, you're cutting into whatever essential functions REM serves. And we're still figuring out exactly what all of those are, but they seem to involve emotional processing, creativity, and certain types of memory consolidation that are different from what happens during slow-wave sleep. This architecture, this nightly cycling through stages, is what sleep researchers call sleep architecture, and it's one of the most important concepts in understanding why sleep quality matters as much as sleep quantity. You could spend 8 hours in bed, but if something is disrupting your ability to cycle through these stages properly, alcohol, for instance, which is notorious for suppressing REM sleep, or sleep apnea, which fragments your sleep by causing you to partially wake up dozens or even hundreds of times per night, then you can get 8 hours of sleep and still feel terrible in the morning. The hours are there, but the architecture is broken. Now, the discovery of REM in 1953 opened the floodgates. Suddenly, sleep researchers had a whole new dimension to explore. And one of the people who did the most to map out the full landscape of sleep stages was a researcher named William Dement. Dement had actually been another student in Kleitman's lab at the University of Chicago. He was there when Oserinsky was making his observations, and he became one of the first people to systematically study the cycling pattern of sleep stages throughout the night. Dement went on to found the first sleep disorders clinic in the world at Stanford University in 1970. And he spent decades advocating for sleep research to be taken seriously as a medical discipline. He's sometimes called the father of sleep medicine, and he was a genuinely charismatic figure, the kind of professor who could fill a lecture hall and keep hundreds of students riveted while talking about something as seemingly mundane as sleep. But Dement's early work in the 1950s and 1960s was crucial for establishing the basic map of sleep architecture that we still use today. He and Kleitman published a landmark paper in 1957 that described the cyclic alternation of REM and non-REM sleep and established the roughly 90-minute cycle length. And Dement also did some of the earliest sleep deprivation experiments, which started to reveal just how essential REM sleep is. When he selectively deprived people of REM sleep by waking them up every time they entered REM but allowing them to sleep otherwise, the results were striking. The subjects became increasingly irritable, anxious, and had difficulty concentrating. And when they were finally allowed to sleep undisturbed, they showed what's called REM rebound. Their brains spent far more time in REM than normal, as if making up for lost time. The brain, it seemed, wasn't just passively cycling through these stages. It was keeping track. It knew what it needed, and if it was deprived, it would compensate. This idea, that the brain actively regulates its own sleep needs, leads us to one of the most elegant concepts in sleep science, and it's something that affects you every single day, whether you're aware of it or not. It's the idea that your desire to sleep, your drive to close your eyes and drift off, isn't governed by just one system. It's governed by two, and they operate independently of each other. And the interaction between them determines almost everything about when you feel alert, when you feel drowsy, and why jet lag feels the way it does. The first system is the circadian rhythm we already talked about, that internal clock ticking away on its roughly 24-hour cycle. But the second system is something different, something that builds up pressure the longer you've been awake, like a spring slowly being compressed hour after hour until the weight of it becomes almost impossible to resist. And that's exactly the right way to think about it, actually. A spring, or maybe a dam filling with water. The longer you stay awake, the more this pressure builds, and eventually it reaches a point where your body is essentially demanding that you sleep. This second system is called the homeostatic sleep drive, and the story of how scientists figured it out is one of those quiet, beautiful pieces of detective work that doesn't get nearly enough attention. So let's talk about what's actually happening in your brain when this pressure builds, because it turns out there's a specific molecule doing most of the heavy lifting, and you interact with it every single morning, whether you know it or not. The molecule is called adenosine. It's a byproduct of cellular energy use. When your neurons are firing, when your brain is working hard to keep you conscious and thinking and navigating your day, it burns through adenosine triphosphate, or ATP, which is basically the energy currency of every cell in your body. And as ATP gets broken down, adenosine accumulates in the spaces between your brain cells, particularly in a region called the basal forebrain. The longer you've been awake, the more adenosine piles up. And adenosine, when it binds to certain receptors on your neurons, has a very specific effect. It makes those neurons less active. It's essentially a dampening signal. It's your brain's way of keeping a running tab on how long it's been since you last slept. Here's the thing that makes this so elegant. Adenosine doesn't care what time of day it is. It doesn't care whether it's light or dark outside. It doesn't know about your circadian rhythm at all. It's purely a function of time spent awake. If you've been up for 16 hours, you've got a lot of adenosine floating around, and you feel sleepy. If you've been up for 4 hours, you don't have much, and you feel alert. It's beautifully simple, and it operates on a completely separate track from that circadian clock in your suprachiasmatic nucleus. And now, here's where it gets really interesting. Think about what happens when these two systems interact, because they're both running at the same time, all the time. And your subjective experience of sleepiness or alertness at any given moment is basically the sum of both signals. So imagine it's 10 o'clock at night. Your circadian clock is winding down, sending signals that it's time to sleep because it's been tracking the light-dark cycle and it knows nighttime is approaching. And simultaneously, your homeostatic drive has been building all day. You've been awake since, say, 7 in the morning, so you've got 15 hours of adenosine accumulation pressing down on your brain. Both systems are saying the same thing: sleep. And that's why 10 at night feels so naturally drowsy for most people. The two systems are aligned. But now, think about what happens with jet lag. Say you fly from New York to London. That's a 5-hour time difference. You land at 7 in the morning London time, but your circadian clock still thinks it's 2 in the morning. It's screaming at you to sleep. But here's the twist. If you manage to sleep on the plane, your homeostatic drive might actually be relatively low. You cleared some of that adenosine. So one system is saying sleep, and the other is saying you're not that tired. And the result is that weird, disoriented, slightly nauseous feeling where you're exhausted, but also weirdly wired. That mismatch between the two systems is essentially what jet lag is. It's not just that your clock is off, it's that your two independent sleep regulation systems are temporarily out of sync with each other, and your brain doesn't quite know what to do with conflicting instructions. The person who formalized this into a proper scientific model was a Swiss sleep researcher named Alexander Borbély. In 1982, he published a paper that laid out what he called the two-process model of sleep regulation. And it's one of those papers that, once you read it, you can't believe nobody put it together sooner. Process S, he called the homeostatic drive, the adenosine buildup, the sleep pressure that accumulates with wakefulness and dissipates during sleep. Process C was the circadian rhythm, that 24-hour oscillation governed by the suprachiasmatic nucleus. And Borbély showed, with mathematical precision, that you could predict when a person would fall asleep, how long they'd sleep, and even the structure of their sleep. How much slow wave versus REM, just by modeling the interaction of these two processes. It was remarkably powerful for such a simple framework. And here's where your morning coffee comes back into the picture. Because caffeine, and this is genuinely one of the most interesting pharmacological stories in everyday life, caffeine works by blocking adenosine receptors. That's it. That's the whole trick. Caffeine doesn't give you energy. It doesn't add anything to your system. What it does is sit in the adenosine receptors like a key that fits the lock but doesn't turn it, preventing the actual adenosine from binding and delivering its sleepiness signal. So all that sleep pressure, all that accumulated adenosine from your hours of wakefulness, it's still there. It's still building up. You just can't feel it, because caffeine is blocking the signal. And this is why the caffeine crash is a real thing. When the caffeine eventually gets metabolized and clears out of those receptors, caffeine has a half-life of about 5 to 6 hours in most adults, by the way, all that adenosine that's been patiently waiting suddenly floods into the receptors all at once, and you feel the full weight of your accumulated sleep pressure hitting you like a wave. You didn't avoid the tiredness. You just postponed it. Now, Borbély's two-process model was published in 1982, and it gave sleep researchers something they'd been lacking, a unified theoretical framework. But it also raised new questions. Because if process S is driven by adenosine and process C is driven by the suprachiasmatic nucleus, what happens when you start messing with one or both of those systems chronically? What happens to people who work night shifts for years? What happens to adolescents whose circadian rhythms naturally shift later? What happens to people who just don't sleep enough, night after night, week after week? And this is where we need to talk about a researcher named David Dingus, because what he did in the late 1990s and early 2000s at the University of Pennsylvania changed our understanding of sleep debt in ways that are still shaping public health policy today. Dingus was interested in a very practical question. How much sleep do people actually need, and what happens when they consistently get less than that? Not total sleep deprivation. Dement and others had already studied that pretty extensively, but the kind of chronic partial sleep restriction that millions of people live with every day. The person who sleeps 6 hours instead of 8, night after night, and insists they're fine. So Dingus and his colleague Hans von Dongen designed an elegantly brutal experiment. They took healthy adults and assigned them to different sleep conditions for 14 consecutive nights. Some got 8 hours in bed, some got 6, some got 4. And a control group was totally sleep-deprived for 3 days straight, just for comparison. Throughout the study, the subjects took regular cognitive tests, reaction time, attention, working memory. And they also rated their own subjective sleepiness. Here's what they found, and it's genuinely unsettling. The people getting 4 hours of sleep per night showed cognitive decline that, after about 6 days, was equivalent to someone who hadn't slept at all for 24 hours straight. Their reaction times tanked. Their attention lapses multiplied. They were making errors at rates that would be dangerous in any real-world context. Driving, operating machinery, making medical decisions. And the 6-hour group? They declined too, just more slowly. By the end of 2 weeks, the 6-hour sleepers were performing at levels comparable to someone who'd been awake for 24 hours straight. 2 weeks of 6-hour nights, and their brains were functioning as if they'd pulled an all-nighter. But here's the kicker, and this is the part that should really make you think. The subjects in the 6-hour group didn't feel that impaired. Their subjective ratings of sleepiness leveled off after a few days. They adapted to feeling a little tired, and then they stopped noticing the decline. But the objective measures, the actual test performance, kept getting worse and worse. In other words, chronic sleep restriction doesn't just impair you, it impairs your ability to recognize that you're impaired. You lose the very faculty you'd need to realize you need more sleep. It's like a carbon monoxide leak for cognition, silent, invisible, and cumulative. This finding, published in 2003, was a bombshell. It meant that the millions of people walking around saying I'm fine on 6 hours might genuinely believe that and be genuinely wrong. Their brains had simply recalibrated their sense of normal. And from an adenosine perspective, what was likely happening is that chronic sleep restriction was preventing the brain from ever fully clearing its adenosine backlog. Each night of insufficient sleep left a residue of sleep pressure, and it accumulated day after day, creating what researchers started calling sleep debt. And sleep debt, it turns out, is not a metaphor. It's a real, measurable neurobiological phenomenon. You can see it in the adenosine levels. You can see it in the EEG patterns. Chronically sleep-restricted people, when they finally get a recovery night, show enormous amounts of slow-wave sleep, their brains gorging on the deepest, most restorative stage as if they'd been starving for it, which, in a very real sense, they had been. Now, this might make you wonder, is there a number? Is there a precise amount of sleep that every human needs? And the honest answer is that it varies, but not as much as people like to think. The research consistently points to somewhere between 7 and 9 hours for adults, with most people needing right around 8. There are genuine short sleepers, people who function well on 6 hours or less, but they're extraordinarily rare, probably less than 1% of the population. And they appear to have specific genetic mutations, particularly in a gene called DEC2, that was identified by Yinghui Fu at the University of California, San Francisco, around 2009. These mutations seem to allow the brain to accomplish its sleep-related maintenance more efficiently, essentially doing 8 hours of work in 6. But for the vast majority of us, that's not the case. If you think you're one of those rare short sleepers, statistically speaking, you're almost certainly not. You're much more likely to be someone whose brain has simply stopped telling you how tired you really are. And this connects to something that Borbély's two-process model predicted, but that took decades to fully appreciate. The way these two systems interact differently at different ages, because your circadian rhythm and your homeostatic drive don't stay constant across your lifespan. They change, sometimes dramatically, and those changes explain some things about sleep that most people find deeply frustrating. Take adolescence, for example. If you've ever had a teenager in your life, or if you remember being one, you know that teenagers seem almost biologically incapable of going to bed at a reasonable hour and then waking up early. And it turns out, that's not laziness, and it's not rebellion. It's physiology. During puberty, the circadian clock shifts later by 1 to 3 hours. The evening rise in melatonin that signals your brain to start preparing for sleep gets delayed. A teenager's brain genuinely isn't ready for sleep at 10 o'clock the way an adult's might be. Their body is telling them midnight, maybe even 1 in the morning. And yet we ask them to be in school by 7:30 or 8, which for their shifted circadian rhythm is the biological equivalent of asking an adult to start work at 4 or 5 in the morning. Mary Carskadon at Brown University has been studying this since the 1990s, and her work has been instrumental in pushing for later school start times. She showed that when adolescents are forced into early schedules that conflict with their shifted circadian rhythms, the result isn't just grumpiness, it's measurable cognitive impairment, increased rates of depression and anxiety, higher rates of car accidents among teen drivers, and even changes in metabolic health. The circadian shift of adolescence is one of the most robust findings in sleep science, and the fact that most school systems still ignore it is one of those places where the gap between what we know and what we do is just staggering, honestly. And it's not like the data is ambiguous. Carskadon's studies, and dozens of others that followed, paint a remarkably clear picture. When school districts have actually tried shifting start times later, even by just 30 or 40 minutes, the results are hard to argue with. One of the most cited examples is the Edina, Minnesota school district, which in 1996 moved its high school start time from 7:25 to 8:30. Researchers tracked the outcomes, and what they found was pretty much exactly what the sleep science predicted. Students reported less daytime sleepiness, fewer depressive symptoms, and, here's a number that gets people's attention, the top 10% of students in terms of academic performance actually saw their SAT scores rise by over 100 points on average, compared to the previous cohort. Now, you can debate the methodology, you can point out confounding variables, and fair enough. But the direction of the effect has been replicated again and again. In 2014, the American Academy of Pediatrics issued a policy statement calling for middle and high schools to start no earlier than 8:30 in the morning, explicitly framing insufficient sleep in adolescents as a public health issue. And yet, as of the early 2020s, the vast majority of American high schools still start before 8:15. Some start before 7:30. California became the first state to legislate later start times in 2019, with a law requiring most high schools to begin no earlier than 8:30, taking effect in 2022. But it remains the exception, not the rule. And the reason I'm spending time on this isn't just because it's an interesting policy debate. It's because it illustrates something fundamental about sleep science as a field. There's this persistent, almost stubborn gap between what we've figured out in the lab and what society actually does with that knowledge. And that gap shows up everywhere, not just in school schedules. It shows up in how we design work shifts, how we staff hospitals, how we train military personnel, how we regulate transportation. Sleep science keeps handing us these findings, and we keep sort of nodding politely and then not changing anything. But let's pull back from the adolescent story for a moment, because there's a bigger thread here about what happens when sleep goes wrong. Not just short-term grogginess, but the deep, systemic consequences of chronic sleep loss on the body. And this is where sleep science starts to intersect with some of the most important medical research of the last few decades. For a long time, sleep deprivation was studied mostly in terms of cognitive performance. Can you stay alert? Can you react quickly? Can you make good decisions? And those studies, like the ones by Dinges and Van Dongen that we talked about earlier, were revelatory. But starting in the late 1990s and accelerating through the 2000s, researchers began uncovering something even more alarming: that chronic sleep loss doesn't just make you foggy, it makes you sick. Like, physically, measurably, progressively sick. One of the landmark studies came from Eve Van Couter's lab at the University of Chicago. In 1999, Van Couter and her colleagues published a study in The Lancet that should have been a wake-up call, no pun intended, for the entire medical community. They took a group of healthy young men, average age around 22, and restricted their sleep to 4 hours per night for 6 consecutive nights. Then, they measured a battery of metabolic and endocrine markers, and the results were startling. After just 6 nights of sleep restriction, these young, healthy subjects showed glucose tolerance levels that were comparable to pre-diabetic patients. Their ability to process sugar from a standardized meal was impaired by about 40%. Their cortisol levels in the evening were elevated, which is the opposite of what you want. Cortisol is supposed to drop at night. And their sympathetic nervous system activity was ramped up, meaning their bodies were stuck in a kind of low-grade stress response. Think about what that means. Take a perfectly healthy 22-year-old, cut their sleep to 4 hours for less than a week, and their metabolic profile starts to resemble someone on the path to type 2 diabetes. Van Couter described it as aging the metabolic system by decades in a matter of days. And when the subjects were allowed to recover, to sleep as much as they wanted for several nights, the markers improved. But the study raised a deeply uncomfortable question. What happens to people who don't recover? What happens to the millions of people who are chronically sleeping 5 or 6 hours a night, year after year? The answer, as subsequent research has shown, is not good. Large epidemiological studies, the kind that follow tens of thousands of people over many years, have consistently found that short sleep duration is associated with increased risk of obesity, type 2 diabetes, cardiovascular disease, and all-cause mortality. The Nurses' Health Study, which tracked over 70,000 women, found that those sleeping 5 hours or less per night had a significantly higher risk of coronary heart disease compared to those sleeping 7 to 8 hours. Similar patterns emerged from the Whitehall 2 study in Britain, which followed over 10,000 civil servants and found that cutting sleep from 7 hours to 5 hours or less was associated with a nearly doubled risk of death from cardiovascular disease. Now, here's where you have to be careful, because epidemiology is tricky. Correlation is not causation, and people who sleep poorly might also be stressed, might also eat badly, might also have underlying health conditions that both disrupt their sleep and damage their cardiovascular system. Researchers know this, and they've spent enormous effort trying to control for confounding variables. And while no single study can definitively prove that short sleep causes heart disease, the consistency of the findings across different populations, different countries, different methodologies, combined with the mechanistic evidence from studies like Van Couter's showing exactly how sleep loss disrupts metabolism and stress hormones, builds a case that's pretty hard to dismiss. And the mechanisms keep getting more detailed. We now know that sleep deprivation increases inflammatory markers like C-reactive protein and interleukin-6. It disrupts the balance of appetite-regulating hormones. Specifically, it decreases leptin, which tells your brain you're full, and increases ghrelin, which tells your brain you're hungry. So when you're sleep deprived, your body is literally sending you stronger hunger signals while simultaneously weakening the signals that would normally tell you to stop eating. If you've ever noticed that you crave junk food after a bad night's sleep, the high-calorie, high-carb stuff, that's not a lack of willpower. That's neurochemistry. Your hypothalamus is responding to altered hormone levels, and functional MRI studies have shown that sleep deprivation amplifies activity in the brain's reward centers in response to food images while dampening activity in the prefrontal cortex, which is the part of your brain responsible for impulse control and rational decision-making. It's almost like your brain is conspiring against you. This is one of those places where sleep science connects to the obesity epidemic in ways that most people don't think about. We tend to frame weight gain as a simple equation: calories in versus calories out, eat less, move more. And there's truth in that, obviously. But when you understand that a third of the adult population is chronically undersleeping, and that undersleeping fundamentally alters the hormonal environment that governs appetite and metabolism, you start to see that the picture is a lot more complicated than just willpower and food choices. Let me tell you about another line of research that emerged around the same time, because it connects to something that affects a huge number of people and went unrecognized for a shockingly long time. I'm talking about obstructive sleep apnea. Now, sleep apnea had been described in the medical literature since the 1960s and 70s. The basic idea is that during sleep, the muscles of the upper airway relax to the point where the airway partially or completely collapses, cutting off airflow. The person stops breathing, sometimes for 10, 20, 30 seconds or more. Blood oxygen levels drop. The brain detects the emergency, triggers a brief arousal, not a full awakening usually, but enough to restore muscle tone and reopen the airway. The person takes a gasping breath, falls back into sleep, and the cycle repeats. In severe cases, this can happen 30, 40, 50 times per hour, every hour, all night long. The person experiencing this usually has no idea it's happening. They might notice that they wake up feeling unrefreshed, that they're excessively sleepy during the day, that they have morning headaches. Their bed partner might notice the snoring, which can be thunderous, and the frightening pauses in breathing. But for decades, sleep apnea was considered a curiosity, something that affected a small number of severely obese older men. It wasn't until the 1990s and 2000s that large-scale epidemiological studies, particularly the Wisconsin Sleep Cohort study led by Terry Young, revealed just how common it actually is. Young's team estimated that roughly 4% of middle-aged men and 2% of middle-aged women had clinically significant sleep apnea, and those numbers have only gone up as obesity rates have risen and diagnostic awareness has improved. Current estimates suggest that somewhere between 15 and 30% of men and 10 to 15% of women have at least mild obstructive sleep apnea, and the vast majority of them are undiagnosed. Here's what makes sleep apnea so insidious, and why it matters for our larger story about sleep and health. Every time the airway collapses and oxygen levels drop, the body mounts a stress response. Cortisol surges, blood pressure spikes, the heart rate jumps, inflammatory markers flood the bloodstream. And this happens dozens of times per hour, night after night, year after year. The cardiovascular system is taking a beating while the person thinks they're resting. Studies have shown that untreated severe sleep apnea roughly doubles the risk of stroke, substantially increases the risk of heart attack, contributes to treatment-resistant hypertension, worsens insulin resistance, and is associated with increased rates of atrial fibrillation. It fragments sleep so thoroughly that the brain never gets the sustained deep sleep and REM sleep it needs for memory consolidation, emotional regulation, and all the restorative processes we talked about earlier. And the treatment, continuous positive airway pressure, or CPAP, which delivers a gentle stream of pressurized air through a mask to keep the airway open, is remarkably effective when people use it. The problem is compliance. A lot of people find the mask uncomfortable, find the noise annoying, find the whole setup undignified. Adherence rates have historically hovered around 50 to 60%, which means a huge number of people who've been diagnosed and given a treatment that works simply don't use it consistently. It's another one of those frustrating gaps between what we know and what we actually do. But sleep apnea research did something important for the broader field of sleep science. It gave doctors a concrete, treatable condition that linked sleep disruption to hard cardiovascular outcomes. It made sleep medicine feel urgent and medical in a way that vague advice about sleep hygiene never quite managed. And it helped build the infrastructure, the sleep labs, the diagnostic protocols, the insurance coding, that allowed sleep medicine to grow as a clinical specialty. Now, while all of this metabolic and cardiovascular research was accumulating, something else was happening in sleep science that would turn out to be just as consequential, maybe even more so. Researchers were beginning to understand what sleep does for the brain itself, not just in terms of alertness and cognitive performance, but in terms of actual physical maintenance. And this story starts with a discovery that sounds almost too strange to be true, involving a plumbing system inside your skull that nobody knew existed until a neuroscientist named Mike Nedergaard figured out what it was doing there. Nedergaard, a Danish researcher working at the University of Rochester Medical Center, had spent years studying glial cells. Those are the brain cells that aren't neurons. For a long time, glia were considered the brain's support staff, the cells that held everything together and cleaned up messes but didn't do anything particularly glamorous. The word glia literally comes from the Greek word for glue. That's how unglamorous they were considered. But Nedergaard had a hunch that glia were doing something far more important than anyone realized. And in 2012, she and her team published a paper that genuinely changed how we think about why we sleep. Here's the setup. Your body has a lymphatic system, a network of vessels that runs alongside your blood vessels and acts as a waste clearance system. It collects cellular debris, metabolic waste products, all the junk that accumulates as your cells go about their business, and it flushes that waste out to be processed and eliminated. It's basically your body's sanitation department. Every organ in your body is connected to this lymphatic network. Every organ except one, the brain. For decades, this was a puzzle. The brain is the most metabolically active organ you have. It uses about 20% of your body's energy, despite being only about 2% of your body weight. All that metabolic activity produces waste, a lot of waste. So where does it go? The brain doesn't have lymphatic vessels. How does it take out the trash? The answer, Nedergaard discovered, is that the brain has its own dedicated waste clearance system, and it works in a way that nobody had predicted. She called it the glymphatic system. Glymphatic, a mashup of glial and lymphatic, because glial cells turn out to be the key players. Here's how it works. Your brain is bathed in cerebrospinal fluid, that clear liquid that surrounds and cushions the brain and spinal cord. What Nedergaard's team showed, using real-time imaging in mice, is that during sleep, cerebrospinal fluid gets actively pumped through the brain tissue along channels that run alongside blood vessels. These channels are formed by the end feet of astrocytes, a type of glial cell, which wrap around blood vessels and create a kind of sleeve, a pathway for the fluid to flow through. The cerebrospinal fluid rushes through these channels, flows between the brain cells, picks up waste products, and then drains out. It's essentially a pressure washing system for your brain. And here's what's really wild about this. The system is dramatically more active during sleep than during wakefulness. Nedergaard's team found that during sleep, the interstitial space in the brain, the gaps between brain cells, expands by about 60%. The cells actually shrink a little, creating wider channels for the fluid to flow through. It's as if the brain streets get wider at night, so the garbage trucks can get through. During waking hours, those streets are narrow and crowded, and the waste clearance system operates at a trickle. During sleep, the floodgates open. Now, you might be thinking, okay, the brain takes out its trash during sleep. That's interesting. But is it really that consequential? And the answer is yes, because of what that trash includes. One of the waste products that the glymphatic system clears is beta amyloid. That name might ring a bell. Beta amyloid is the protein that accumulates in the brains of people with Alzheimer's disease, forming the characteristic plaques that are one of the hallmarks of the condition. Nedergaard's work showed that the glymphatic system clears beta amyloid from the brain about twice as fast during sleep as during wakefulness. Think about what that means. Every night, while you're lying there apparently doing nothing, your brain is actively flushing out a protein that, if it accumulates, is associated with one of the most devastating diseases of aging. And this immediately raised a question that researchers are still working on today. Does chronic sleep deprivation, by reducing the brain's opportunity to clear beta amyloid, contribute to the development of Alzheimer's disease? The epidemiological evidence is suggestive. Multiple studies have found that people who consistently sleep less than 6 hours a night have a significantly higher risk of developing Alzheimer's later in life. A 2017 study from Washington University in St. Louis, led by David Holtzman and colleagues, showed that even a single night of sleep deprivation increased beta amyloid levels in the cerebrospinal fluid of healthy human volunteers. And a study using PET imaging showed that just one night of total sleep deprivation led to a significant increase in beta amyloid accumulation in the human brain, particularly in the hippocampus and thalamus, regions that are important for memory and that are vulnerable in early Alzheimer's. Now, it's crucial to be careful here. Correlation isn't causation, and Alzheimer's disease is complex, with genetic, environmental, and lifestyle factors all playing roles. It's also possible that the relationship runs in both directions, that early Alzheimer's pathology disrupts sleep, which then accelerates further amyloid accumulation, creating a vicious cycle. Some researchers think that's exactly what happens, but the glymphatic discovery gave the field something it had been missing: a plausible biological mechanism linking sleep loss to neurodegeneration. It wasn't just that sleep-deprived people performed worse on memory tests. There was now a physical hydraulic explanation for why the brain needs sustained periods of unconsciousness. It needs time to clean itself. Nedergaard's work also revealed that the glymphatic system is most active during deep non-REM sleep. Those slow, rolling delta waves we talked about earlier, the ones that dominate the first few hours of the night. This makes a kind of intuitive sense. During deep sleep, neural activity is highly synchronized. Millions of neurons firing and falling silent together in those big, slow oscillations. Some researchers have suggested that these coordinated waves of activity and silence actually help drive the pulsing of cerebrospinal fluid through the brain, like a slow, rhythmic pump. The very brain waves that characterize deep sleep might be mechanically involved in waste clearance. That's an idea that's still being tested. But if it holds up, It means that the electrical signature of deep sleep isn't just a marker of a particular brain state. It's functionally important for brain maintenance in a very physical, almost plumbing-like way. And this connects back to something we touched on earlier about aging. As people get older, they tend to get less deep sleep. The slow-wave activity that dominates the first sleep cycles in young adults diminishes significantly with age. By the time someone is in their 60s or 70s, they might be getting a fraction of the deep sleep they got in their 20s. If the glymphatic system depends on deep sleep to operate at full capacity, then the age-related decline in deep sleep might mean a progressive decline in the brain's ability to clear waste, including beta amyloid, and that timeline matches up uncomfortably well with the typical timeline for Alzheimer's development, which involves decades of slow amyloid accumulation before symptoms appear. Matthew Walker, the neuroscientist at UC Berkeley who wrote the best-selling book Why We Sleep, has been one of the most vocal advocates for taking this connection seriously. Walker's own research has focused on how sleep deterioration in older adults relates to memory decline and Alzheimer's pathology, and he's argued that improving sleep quality in middle age might be one of the most promising strategies for reducing Alzheimer's risk. It's a compelling argument, though it's worth noting that some of Walker's specific claims have been scrutinized and debated within the field. The relationship between sleep and Alzheimer's is real and supported by evidence, but the exact magnitude of the risk and the degree to which improving sleep can actually prevent the disease are still being worked out. Science is messy like that. The broad strokes are clear, but the fine details take decades to resolve. So let's step back for a moment and think about where we are in this story. By the mid-2000s, sleep science had built a remarkably detailed picture. We understood the architecture of sleep, the stages, the cycles, the brain waves. We understood the two-process model, the homeostatic drive and the circadian clock working together. We understood what happens when you don't get enough sleep, the cognitive impairment, the metabolic disruption, the cardiovascular risk, the immune suppression. And now, with the glymphatic system, we had a compelling new reason to take sleep seriously. The brain literally needs sleep to clean itself, and failing to do so might contribute to neurodegeneration. The case for sleep had never been stronger. And yet the world kept getting worse at sleeping. This is the part of the story that's both fascinating and a little maddening, because while sleep scientists were accumulating all this evidence about why sleep matters, the modern world was engineering sleep out of people's lives with remarkable efficiency. And to understand how that happened, you have to think about light. Not sunlight, artificial light. Specifically, the kind of light that comes from screens. Because the story of sleep in the 21st century is inseparable from the story of how we flooded our evenings with a particular wavelength of light that our circadian system interprets as a signal to stay awake. Remember the suprachiasmatic nucleus, that tiny cluster of cells in the hypothalamus that acts as the master clock? The SCN sets its timing primarily through light signals received from the retina. But here's the thing. The cells in the retina that communicate with the SCN aren't the rods and cones you learned about in biology class, the ones responsible for vision. In the early 2000s, researchers discovered a third type of photoreceptor in the retina called intrinsically photosensitive retinal ganglion cells, or IPRGCs. These cells contain a light-sensitive pigment called melanopsin, and they don't contribute much to conscious vision. Instead, they send signals directly to the SCN, telling the master clock about the ambient light environment. And melanopsin is most sensitive to light in the short wavelength range, blue light around 480 nanometers. This is where the story gets uncomfortable, because blue light is exactly the kind of light that LED screens emit in abundance. Your phone, your tablet, your laptop, your television, they all produce significant amounts of short wavelength blue light. And when that light hits the melanopsin-containing cells in your retina in the evening hours, it sends a signal to your SCN that says, essentially, it's still daytime. The SCN responds by suppressing the release of melatonin from the pineal gland. Melatonin, you'll recall, is the hormone that signals the body that nighttime has arrived. It doesn't knock you out, but it opens the gate for sleep, telling your physiology to start the transition toward rest. When blue light from screens suppresses melatonin in the evening, it delays that transition. Your biological night gets pushed later. Charles Czeisler at Harvard, one of the leading circadian researchers in the world, has been particularly vocal about this issue. His lab published studies showing that reading on a light-emitting device before bed compared to reading a printed book suppressed melatonin secretion, delayed the circadian clock, reduced the amount of REM sleep, and left people feeling sleepier the next morning, even after 8 hours in bed. The effect wasn't subtle. Melatonin onset was delayed by about an hour and a half, and this wasn't from some industrial light source. This was from an iPad at normal brightness, held at a normal reading distance, used for a few hours before bed. The kind of thing millions of people do every single night without thinking twice about it. And the timing of this research matters. The iPhone launched in 2007, the iPad in 2010. By the mid-2010s, smartphones were ubiquitous, and the average American was spending hours each evening bathed in screen light. The devices arrived faster than the science could warn people about what the light was doing to their circadian systems. And even once the warnings came, changing behavior proved incredibly difficult, because the devices weren't just light sources. They were portals to social connection, entertainment, news, work. The light was almost incidental to why people used them, but it was the light that was doing the biological damage. Now, the technology industry did respond, eventually. Apple introduced night shift mode in 2016, which reduces blue light emission from screens in the evening. Android followed with similar features. But the research on whether these features actually help is mixed. Some studies suggest that reducing blue light from screens has only a modest effect on melatonin suppression, partly because the overall brightness of the screen still matters, and partly because the stimulating content on the screen, the social media feeds, the news alerts, the one more episode pull of streaming video, keeps the brain aroused independently of the light itself. It turns out that the problem with screens before bed isn't just photonic, it's psychological. The light shifts your clock, but the content keeps your mind racing, and both of those things work against the kind of quiet, gradual wind down that your brain needs to transition from wakefulness into sleep. And this is something that researchers have started paying a lot more attention to in recent years. The idea that what you're doing with a screen matters just as much as the light coming off of it, maybe even more. Think about what happens when you're scrolling through social media at 11 at night. Every few seconds, there's something new. A photo, a comment, a headline, a notification. Each one of those is a tiny little hit of novelty, and your brain responds to novelty. It's wired to. The dopamine system lights up, not in some massive surge, but in these small, repeated pulses that keep you engaged, keep you alert, keep you reaching for the next scroll. And that's exactly the opposite of what your brain needs to be doing in the hour before sleep. It needs to be ramping down, reducing arousal, letting the default mode network take over, that quiet, diffuse, slightly wandering state of mind that naturally precedes sleep onset. But instead, you're feeding it a constant stream of microstimulation, and it stays locked in an attentive, reactive mode. There was a study out of Norway, published around 2014 by Mari Heising and colleagues, that looked at screen use in nearly 10,000 adolescents. And what they found was striking, but maybe not surprising. The more time teens spent on electronic devices before bed, the longer it took them to fall asleep, the less total sleep they got, and the more likely they were to report sleep deficiency. And the relationship was dose-dependent, meaning it wasn't just about whether you used a screen or not. It was about how much. An hour of screen time before bed was worse than 30 minutes, and 2 hours was worse than 1. But here's the part that's really interesting. The type of device mattered. Computers and smartphones were more strongly associated with sleep disruption than television, even though a TV screen is typically much larger and brighter. Why? Probably because of interactivity. A TV show just plays. You sit there passively. But a smartphone demands your active engagement. You're tapping, scrolling, responding, choosing. Your brain is working. And working brains don't fall asleep easily. This connects to something sleep researchers call cognitive arousal, which is distinct from physiological arousal. Your heart rate might be fine. Your muscles might be relaxed, but your mind is spinning. And cognitive arousal is one of the most common complaints among people with insomnia. They'll tell you their body is tired, but their brain won't shut off. Pre-sleep screen use, especially the interactive kind, seems to prime exactly that state. Now, there's another dimension to this that doesn't get talked about enough, and it has to do with emotional valence, the emotional tone of what you're consuming. If you're reading distressing news before bed, or getting into an argument in a comment section, or comparing your life to the curated highlight reels on Instagram, you're not just cognitively aroused, you're emotionally activated. Stress hormones like cortisol can spike, and cortisol is essentially the anti-sleep hormone. It promotes wakefulness, alertness, vigilance. It's exactly what you want at 8 in the morning and exactly what you don't want at 11 at night. So the content on the screen can actually trigger a hormonal response that directly opposes sleep onset. And all of this, the light, the cognitive stimulation, the emotional activation, compounds, they don't just add up, they interact. The blue light delays your melatonin. The stimulating content keeps your cortex firing. The emotional charge elevates your cortisol. And together, they create a state that is profoundly incompatible with the gentle, gradual slide into unconsciousness that sleep is supposed to be. It's like trying to land a plane while someone keeps pulling back on the throttle. You can't descend if the engines are still pushing you up. So what actually works? What does good sleep hygiene look like in a world saturated with screens and notifications and the constant pull of digital engagement? Well, this is where we get into the practical side of sleep science, and it's worth spending a little time here because the research has actually converged on some pretty clear recommendations, even if following them is easier said than done. The concept of sleep hygiene itself goes back to Peter Howry, a sleep researcher at the Mayo Clinic who coined the term in the late 1970s. His original guidelines were straightforward: keep a regular sleep schedule, avoid caffeine and alcohol close to bedtime, make your bedroom dark and cool. Don't use your bed for anything other than sleep and intimacy. These recommendations were based on clinical observation as much as controlled research, but over the decades, they've been validated and refined. And while no single hygiene practice is a magic bullet, the cumulative effect of following several of them is genuinely significant. Temperature, for instance. This one is fascinating because it ties directly back to the circadian system we talked about earlier. Your core body temperature follows a daily rhythm. It peaks in the late afternoon and drops through the evening, reaching its lowest point in the early hours of the morning, usually around 4 or 5 a.m. And that drop in core temperature isn't just correlated with sleep onset. It actually helps trigger it. Your brain uses the falling temperature as a cue that it's time to sleep. This is why a warm bath before bed can paradoxically help you sleep. Not because the warmth relaxes you, though it might, but because when you get out of the bath, your dilated blood vessels rapidly shed heat. Your core temperature drops faster than it otherwise would, and that accelerated cooling signals your brain that sleep is imminent. Researchers at the University of Texas published a meta-analysis in 2019. Shahab Hagaya was the lead author, looking at over 5,000 studies, and they found that a warm bath or shower 1 to 2 hours before bed, at around 40 to 43 degrees Celsius, was associated with significantly faster sleep onset, about 10 minutes faster on average. That might not sound like much, but for someone who routinely lies awake for 30 or 40 minutes, shaving 10 minutes off that is meaningful. And the bedroom temperature itself matters. Most sleep researchers recommend keeping the room cool, around 65 to 68 degrees Fahrenheit, or about 18 to 20 Celsius. That feels cold to a lot of people, but remember, your body is trying to dump heat as part of the sleep initiation process. If the room is too warm, your body can't offload that heat efficiently, and you end up stuck in a state of thermal discomfort that fragments your sleep, even if you don't fully wake up. You cycle into lighter stages more often, you get less deep NREM, and you wake up feeling unrested even after what seemed like a full night. Light control is equally important, and this goes beyond just screens. The ambient light in your home in the hours before bed matters. Researchers like Joshua Gooley at Harvard have shown that ordinary room lighting, the kind you'd have in a well-lit living room, can suppress melatonin by about 50% compared to dim light conditions. 50%. That's not from a screen 6 inches from your face. That's just from having the lights on in your house. And this is where the modern built environment works against us, because our homes are flooded with artificial light from the moment the sun goes down until the moment we switch everything off and climb into bed. There's no gradual dimming, no twilight equivalent. It's full brightness and then darkness, and your circadian system doesn't get the slow ramp down it evolved to expect. Some people have started using dimmer switches or switching to warmer, lower wattage bulbs in the evening, or even using candlelight or salt lamps in the hour before bed. And the research supports this, reducing light exposure in the evening, particularly in the blue-enriched spectrum, helps preserve the natural rise of melatonin and supports earlier, easier sleep onset. It's a simple intervention, but it requires a kind of intentionality that goes against the grain of how most of us live. Regularity might be the single most underrated factor. Matthew Walker and others have emphasized this repeatedly. Keeping a consistent wake time and bedtime, even on weekends, is one of the most powerful things you can do for your sleep quality. And the reason ties back to Borbély's two-process model. Remember, your sleep drive and your circadian rhythm need to be aligned for sleep to work well. If you shift your schedule by 2 or 3 hours on the weekend, sleeping in until noon on Saturday, then trying to fall asleep at 10 on Sunday night, you're essentially giving yourself social jet lag. Your circadian clock hasn't shifted just because your alarm didn't go off. You're asking your body to fall asleep at a time when your internal clock thinks it's still early evening, and the result is that Sunday night insomnia that so many people experience. It's not anxiety about Monday morning, or not entirely. It's a circadian misalignment that you created by sleeping in. Till Roenneberg, a chronobiologist at Ludwig Maximilian University in Munich, actually coined the term social jet lag around 2006, and his research showed that the discrepancy between people's weekday and weekend sleep schedules was associated with higher BMI, increased use of stimulants, and worse overall health outcomes. The bigger the gap between your weekday and weekend sleep timing, the worse the effects. And this was independent of total sleep duration, meaning it wasn't just about getting less sleep. It was about the irregularity itself. This brings up something important about individual variation, because not everyone's circadian clock runs on the same schedule. You've probably heard the terms morning lark and night owl, and these aren't just personality labels. They reflect genuine biological differences in circadian timing, what researchers call chronotype. Some people's internal clocks naturally run a bit early, so they feel alert in the morning and sleepy by 9 or 10 at night. Others run late. They don't hit their stride until the afternoon and don't feel genuinely sleepy until midnight or later. And this isn't laziness or discipline, it's genetics. Studies of clock gene variants have shown that chronotype is substantially heritable, and forcing a natural night owl to conform to an early bird schedule is fighting biology. The problem is that society is largely structured around early chronotypes. School starts early. Most jobs start early. The cultural narrative valorizes the early riser. The early bird gets the worm, and stigmatizes the person who sleeps until 10 as lazy or unmotivated. But if your circadian clock is set 2 hours later than average, waking at 6 a.m. is biologically equivalent to someone else waking at 4 a.m. You're not getting less motivated sleep. You're getting less sleep, period, because your clock won't let you fall asleep early enough to get a full night before that alarm goes off. And this has real consequences. Roenneberg's data showed that late chronotypes living in an early start world accumulated chronic sleep debt, had higher rates of depression, and were more likely to smoke and drink, not because being a night owl is inherently unhealthy, but because the mismatch between their biology and their schedule created a state of perpetual circadian stress. When late chronotypes were allowed to follow their natural schedule, sleeping from, say, 1 a.m. to 9 a.m., their health outcomes improved. The sleep itself wasn't the problem. The timing was. This is where the conversation about sleep starts to shade into questions about how we design our institutions, our workplaces, our cities. Because if we know that a significant portion of the population, maybe 20 to 25%, are natural late chronotypes, and we know that forcing them into early schedules damages their health, then the way we structure work and school hours becomes a public health issue, not just a personal preference. Some companies have started experimenting with flexible start times, and the results have been encouraging. Better employee well-being, lower absenteeism, even improved productivity. It turns out that letting people work when their biology says they're most alert produces better outcomes than forcing everyone into the same 8 to 5 box. But flexible scheduling is a luxury that many workers simply don't have, especially in shift work, which is where some of the most dramatic sleep-related health consequences show up. And shift work is worth talking about in some detail because it represents the most extreme version of what happens when you systematically override your circadian system, and the data on what it does to the human body over time is frankly sobering. The International Agency for Research on Cancer, which is part of the World Health Organization, classified night shift work as a probable carcinogen in 2007. Let that sink in for a second. Not a possible carcinogen, a probable one. That puts it in the same category as certain industrial chemicals. And they didn't make that classification lightly. It was based on decades of accumulated epidemiological evidence showing that long-term shift workers had elevated rates of breast cancer, prostate cancer, colorectal cancer, and cardiovascular disease that couldn't be fully explained by lifestyle factors alone. The numbers are striking. A meta-analysis published in the British Medical Journal in 2012, looking at more than 2 million workers across multiple studies, found that shift workers had a 23% increased risk of heart attack, a 5% increased risk of stroke, and a 24% increased risk of coronary events overall. And the risk scaled with duration. Someone who'd been doing rotating night shifts for 15 or 20 years faced substantially higher risk than someone who'd done it for 3. This wasn't about one bad night. This was about years and years of telling your body it was daytime when every cell in your system knew it was dark outside. And here's what's really important to understand about why shift work is so damaging. It's not just sleep deprivation, though that's certainly part of it. It's circadian misalignment. Remember the suprachiasmatic nucleus, that tiny cluster of neurons we talked about earlier, the one that acts as your master clock. When you work a night shift, you're fighting that clock at every level. Your SCN is still receiving light cues during the day, still trying to run its normal program. But you're asking your body to be alert and functional at 3 in the morning when your core temperature is at its lowest, your melatonin is peaking, and your cognitive processing speed is in the basement. And then you're asking it to sleep at noon when cortisol is elevated, ambient light is flooding through your curtains, and the neighborhood is alive with lawnmowers and delivery trucks and all the noise of a world that's wide awake. The result is that most shift workers never fully adapt. Studies have shown that even permanent night workers, people who work nights for years, rarely achieve complete circadian realignment. Their melatonin rhythms stay partially anchored to the solar day. They exist in a kind of biological no man's land, not fully adjusted to either schedule. And rotating shift workers have it even worse, because just as their body starts to partially adjust to nights, their schedule flips again, and they're back on days, and the whole miserable process of readjustment starts over. A researcher named Diane Boyven at McGill University did some really elegant work on this in the early 2000s. She brought shift workers into the lab and measured their circadian markers, melatonin, cortisol, core body temperature, across multiple shift rotations. What she found was that the degree of adaptation varied enormously between individuals, but on average, even after a week of consecutive night shifts, most workers had only partially shifted their circadian phase. Their biology was in a state of chronic compromise. And this matters because circadian alignment affects virtually every physiological system: immune function, glucose metabolism, hormone regulation, DNA repair. When those processes are running on the wrong schedule, errors accumulate. Cells divide when they shouldn't. Inflammatory markers stay elevated. Insulin sensitivity drops. The body's maintenance routines, which are timed to occur during sleep, get disrupted or truncated. This connects back to something we touched on earlier when we were talking about Mike Niedergard's glymphatic system. Remember how the brain's waste clearance mechanism is most active during deep sleep? Well, shift workers consistently get less deep sleep than day workers. Their sleep is shorter, more fragmented, and architecturally different. Less slow-wave sleep, more time in lighter stages, which means their brains may not be clearing metabolic waste as effectively. And while the long-term implications of that are still being studied, the connection to neurodegenerative risk is one that researchers are watching very closely. Now, you might be wondering, if shift work is so harmful, why do we do it? And the answer, of course, is that modern society can't function without it. Hospitals need nurses at 3 a.m. Power plants need operators around the clock. Airports, police departments, fire stations, factories, data centers. The infrastructure of contemporary life runs 24 hours a day, and someone has to be there. About 15 to 20% of the workforce in most industrialized countries does some form of shift work. That's tens of millions of people in the United States alone. So the question becomes not whether to eliminate shift work. That's not realistic, but how to make it less destructive. And this is where sleep science has actually started to have a practical impact. Researchers like Charles Czeisler at Harvard, who we mentioned earlier in the context of light and melatonin, have spent decades working with industries to design better shift schedules. One of the key findings is that the direction of shift rotation matters. Forward rotating schedules, where you move from days to evenings to nights, are easier on the body than backward rotating ones, because they work with the natural tendency of the circadian clock to drift slightly later. It's the same reason it's easier to stay up late than to go to bed early. Your clock would rather extend the day than compress it. Czeisler's team worked with a police department in Philadelphia in the early 1990s and redesigned their shift schedule based on circadian principles. Longer rotation periods, forward rotation, and strategic use of bright light exposure during night shifts to help nudge the circadian clock toward adaptation. The results were meaningful. Officers on the new schedule slept more, reported better mood, had fewer on-the-job accidents, and, here's a detail I love, fell asleep less often while driving home after their shift. That last one is not trivial. Drowsy driving after a night shift is one of the most dangerous consequences of shift work, and it kills people every year. The strategic use of light is actually one of the most promising tools for helping shift workers. If you can expose someone to bright, blue-enriched light during the first half of their night shift, and then have them wear dark or amber-tinted glasses on their drive home, and sleep in a completely darkened room, you can accelerate circadian adaptation significantly. It doesn't solve the problem entirely, but it moves the needle. Some forward-thinking employers have started installing bright light panels in break rooms and work areas for night shift employees. It's a relatively low-cost intervention with real physiological benefits. Melatonin supplementation is another tool, though it has to be timed correctly, which is trickier than most people realize. Taking melatonin isn't just about making yourself sleepy. It's about sending a timing signal to your circadian system. Taken at the right point in your circadian cycle, it can help shift your clock in the desired direction. Taken at the wrong time, it can actually make the misalignment worse. This is why over-the-counter melatonin use, without understanding the underlying circadian principles, often produces inconsistent results. The dose matters less than the timing, which is counterintuitive for most people who think of it as a simple sleeping pill. But let's zoom out for a moment, because shift work is really just the most extreme case of a broader phenomenon that affects almost everyone to some degree. And that phenomenon is the fundamental tension between biological time and social time. Roenneberg called it social jet lag, and we touched on it earlier. But it's worth sitting with the idea a little longer because it's so pervasive. Almost everyone in modern society experiences some version of it. You stay up later on weekends than on weekdays. You sleep in on Saturday morning. Then Sunday night, you can't fall asleep because your clock has drifted. And Monday morning, the alarm drags you out of bed before your body is ready. That weekly oscillation, even if it's just an hour or two, creates a mild but chronic form of circadian disruption. And Roenneberg's research suggested that the magnitude of your social jet lag predicted health outcomes. People with larger weekend-to-weekday sleep discrepancies had higher BMI, were more likely to be smokers, and reported lower well-being. Think about what that means. It's not just about how much you sleep. It's about the regularity of when you sleep. And this brings us to one of the more recent and genuinely exciting developments in sleep research: the work on sleep regularity as an independent health predictor. A study published in 2023 by Daniel Buissy and colleagues using data from the UK Biobank, which is this enormous dataset of nearly 90,000 participants wearing accelerometers, found that sleep regularity was a stronger predictor of all-cause mortality than sleep duration. Let me say that again, because it's remarkable. How consistently you went to bed and woke up at the same time predicted your risk of dying better than how many hours you slept. People with highly irregular sleep patterns had significantly higher rates of cardiovascular disease, metabolic disorders, and cancer, even after controlling for total sleep time and a host of other variables. This is one of those findings that reshapes how you think about the whole topic. For years, the public health message about sleep has been focused almost entirely on duration. Get your 7 to 9 hours. And duration matters absolutely. But regularity might matter just as much, or even more. Your circadian system thrives on consistency. It's a prediction machine, remember? It anticipates when sleep is coming and begins preparing your body accordingly. When you go to bed at the same time every night, your melatonin rises on schedule, your core temperature drops on cue, your cortisol withdrawal happens at the right moment. Everything is synchronized. But when your schedule is erratic, the system can't predict, and those preparatory processes get mistimed or blunted. This has practical implications that are actually kind of liberating. Because while you can't always control how long you sleep, life is complicated, babies wake up, deadlines loom, you can often control when you go to bed. And simply anchoring your schedule, keeping your wake time consistent even on weekends, even if it means you occasionally get a shorter night, may do more for your long-term health than obsessing over hitting exactly 8 hours. It's the steadiness that your body craves. And this idea that rhythm and regularity are as important as quantity brings us to something that's been quietly transforming the field over the last decade or so. Because researchers have started to realize that sleep isn't just one monolithic thing you either get enough of or don't. It's a complex, multidimensional behavior with duration, timing, regularity, architecture, and quality, all contributing independently to health outcomes. And measuring all of those dimensions simultaneously has only become possible because of a technology that barely existed 20 years ago, but is now strapped to the wrists of hundreds of millions of people around the world. Consumer wearable devices, fitness trackers, smartwatches have created a flood of sleep data unlike anything researchers have ever had access to, and what they're finding in that data is reshaping how we think about sleep at a population level. Because for most of the history of sleep science, going all the way back to Berger's first EEG recordings in the 1920s. Researchers had a fundamental problem. They could study sleep in extraordinary detail, but only in tiny numbers. A polysomnography study might have 30 participants. A really big one might have a few hundred. And every single one of those people had to come into a lab, get wired up with electrodes, and sleep in an unfamiliar environment while being watched, which, as you can imagine, doesn't exactly produce the most natural sleep. Researchers called it the first night effect. People almost always sleep worse on their first night in a lab, and sometimes the second night too. So the data was precise but artificial, and it came from small, often unrepresentative samples. Usually young, healthy college students, because those are the people who show up for sleep studies. Then along came actigraphy, wrist-worn devices that measured movement to estimate sleep and wake patterns. These had been around since the 1970s and 80s, and they were useful because you could send someone home wearing one for a week or two and get a rough picture of their natural sleep schedule. But they were expensive, clunky, and mostly stayed within the research world. What changed everything was when companies like Fitbit, founded in 2007, and later Apple, Garmin, Samsung, and others, started putting accelerometers and heart rate sensors into consumer devices that millions of people were already wearing for other reasons: step counting, exercise tracking, notifications. Suddenly, almost by accident, there was an enormous distributed sleep monitoring network generating data every single night, across every age group, every time zone, every season. Now here's the thing you need to understand about this data. It's not polysomnography. A wrist-worn device can't tell you exactly when someone enters stage 3 deep sleep, or when a REM episode begins. It can't measure brain waves. What it can do, with increasingly impressive accuracy, is estimate when you fell asleep, when you woke up, how many times you were restless during the night, and, using heart rate variability and movement patterns together, make reasonable inferences about sleep stages. The algorithms have gotten better year after year, and while no serious sleep scientist would use a consumer wearable as a diagnostic tool, the sheer scale of the data compensates for a lot of the individual imprecision. When you have sleep data from tens of millions of people across years, patterns emerge that no laboratory study could ever reveal. One of the first really striking findings came from a 2016 study led by Daniel Forger and Olivia Walsh at the University of Michigan. They built a smartphone app called Entrain, originally designed to help people adjust to new time zones, and they collected data from users in over 100 countries. What they found was fascinating. Bedtimes varied enormously across cultures. People in Japan and Singapore went to bed latest, people in Australia and New Zealand earliest. But wake times were much more similar worldwide. This suggested that social pressures, things like work schedules, school, commuting, tend to dictate when people wake up. And that variation in sleep duration is mostly driven by when people choose to go to bed, which connects beautifully back to what Roenneberg found with social jet lag. Societies aren't depriving people of sleep by making them get up too early, so much as by creating evening environments that keep people up too late. The compression happens from the bedtime side, and then the really massive data sets started arriving. Fitbit published aggregated data from billions of nights of sleep across millions of users, and the patterns were remarkably consistent with what lab studies had suggested, but could never prove at scale. Sleep duration really does decline with age, but not as dramatically as people think. The big drop happens between your teens and your 30s, and then it plateaus. Women consistently sleep slightly longer than men, but report worse sleep quality, which is an interesting paradox that probably reflects hormonal differences, higher rates of insomnia, and the fact that women are more likely to have their sleep disrupted by caregiving. People sleep more on weekends than weekdays, with the gap being largest for young adults. That social jet lag signature showing up crystal clear in the data. But here's what's really clever about what researchers started doing with this information. They began linking wearable sleep data to health outcomes at a scale that was previously impossible. BICE's UK Biobank work that we touched on, showing that sleep regularity predicted mortality better than duration, was part of this wave. And similar findings started coming from other directions. A 2020 study using data from over 60,000 people wearing Fitbit devices found that higher heart rate variability during sleep, which is a marker of good autonomic nervous system function and typically reflects deeper, more restorative sleep, was associated with fewer self-reported sick days. People whose wearable data showed more consistent sleep-wake patterns had better next-day heart rate recovery after exercise. The correlations were modest individually, but they were consistent and they pointed in the same direction. Regularity and quality matter, and now we could actually see them in real time. This flood of data has also started revealing things about sleep architecture that we couldn't appreciate before. For instance, the proportion of REM sleep you get appears to change not just with age, but with your sleep timing relative to your chronotype. Night owls forced onto early schedules don't just get less total sleep, they get proportionally less REM sleep because REM is concentrated in the later cycles of the night, and those are exactly the cycles that get cut short when your alarm goes off too early. This was suspected from lab work, but seeing it confirmed across hundreds of thousands of real-world nights was powerful. Now, there's a flip side to all this data, and it's worth talking about because it connects to something genuinely important about how we relate to sleep. In 2017, a group of researchers at Rush University Medical Center in Chicago, led by Kelly Barron and a clinical psychologist named Sabra Abbott, published a paper describing a new phenomenon they called orthosomnia. The word is modeled on orthorexia, the unhealthy obsession with eating only healthy food, and orthosomnia describes people who become so fixated on optimizing their sleep tracker data that the anxiety about their sleep actually makes their sleep worse. They'd see patients who slept fine by every objective measure, but were convinced they had a problem because their watch told them they only got 14% deep sleep instead of the 20% they'd read was ideal. People were lying in bed longer trying to boost their sleep scores, which is actually counterproductive. It leads to more fragmented sleep and can trigger insomnia. The tracker, meant to help, was becoming a source of stress. And this is a genuinely important lesson, maybe one of the most important ones in this whole story. Because sleep is one of those things that gets worse when you try too hard. It's paradoxical. The more you monitor it, worry about it, optimize it, the more elusive it can become. Sleep researchers have a term for this. They call it sleep effort, and it's one of the strongest predictors of chronic insomnia. The people who try hardest to sleep are often the people who sleep worst. This is why cognitive behavioral therapy for insomnia, CBTI, which is now considered the gold standard first-line treatment ahead of medication, works the way it does. It doesn't give you techniques to force yourself to sleep. Instead, it dismantles the anxiety and the counterproductive behaviors that are keeping you awake. Things like spending too long in bed, clock watching, catastrophizing about tomorrow's performance if you don't fall asleep right now. CBT1 was developed over decades with major contributions from researchers like Arthur Spielman, who proposed the 3P model of insomnia: predisposing, precipitating, and perpetuating factors, and Charles Morin at Laval University in Quebec, who ran some of the largest clinical trials showing that CBTI outperforms sleeping pills, not just in the short term, but especially in the long term, because it addresses root causes rather than masking symptoms. So there's this beautiful tension in modern sleep science. On one hand, we know more than ever about what good sleep looks like, why it matters, and what disrupts it. On the other hand, the healthiest relationship with sleep might be one where you don't think about it too much, where you set up the conditions: consistent schedule, dark room, cool temperature, limited late-night screen exposure, and then you let go. You trust the system, because your body already knows how to sleep. It's been doing it since before you were born, literally. Fetuses spend most of their time in sleep-like states. The machinery is ancient and robust. What it needs from you isn't micromanagement. It needs you to stop getting in its way. And maybe that's the thread that ties this whole story together, if you think about it. From the very beginning, from the ancient Greeks wondering whether sleep was caused by vapors rising from the stomach, to Berger quietly recording the first human brain waves in his Gina laboratory, to Oserinsky noticing his son's eyes darting beneath closed lids, to Kleitman spending 32 days in a Kentucky cave, to Dement founding the first sleep clinic, to Borbély sketching his elegant two-process model on a napkin. The entire history of sleep science has been a long, slow process of learning to respect something we'd been taking for granted. Every major discovery has essentially been a variation on the same theme. Sleep is doing more than you thought. It's more complex than you assumed. It's more important than you realized. And the systems governing it are more sophisticated than anyone imagined. The suprachiasmatic nucleus, that tiny cluster of neurons smaller than a grain of rice, keeping time for your entire body. The glymphatic system, washing your brain clean every night like a dishwasher running its cycle. The adenosine buildup, that quiet chemical hourglass measuring how long you've been awake. The melatonin signal, translating darkness into drowsiness. The intricate choreography of sleep stages cycling through the night, each one doing different work, consolidating different types of memory, releasing different hormones, restoring different systems. It's all happening right now, or it will be soon, as you lie here and let your mind drift. And the researchers who uncovered all of this, they were mostly people who stumbled into the field almost by accident. Kleitman started out studying sleep deprivation because nobody else thought it was interesting enough to bother with. Oserinsky was a graduate student who almost dropped out. Dement wandered into Kleitman's lab looking for a thesis topic and ended up spending 60 years mapping the landscape of sleep. Rechtschaffen, Jouvet, Karskadon, Cheisler, Borbély, Nedergaard, none of them set out to revolutionize medicine. They just followed their curiosity, one question at a time, one night at a time, and the answers kept being bigger and stranger and more consequential than anyone expected. That's the thing about sleep science. It keeps surprising us. And it will keep surprising us, because there's still so much we don't understand. We don't fully know why we dream or what dreams are for, despite a century of theories. We don't know exactly how sleep consolidates memory at the molecular level. We have strong clues involving synaptic homeostasis and memory replay, but the full picture remains fuzzy. We don't know why some people are resilient to sleep deprivation and others fall apart after one bad night. We don't know whether the glymphatic system can be enhanced or whether doing so could actually prevent Alzheimer's disease. These are open questions, and they're being worked on right now, tonight, in labs around the world, by people who are probably not getting enough sleep themselves. But you don't need to worry about any of that right now. You've heard the story, or as much of it as you needed to hear. The history, the science, the people, the discoveries. And the beautiful thing is that your body doesn't need you to understand any of it in order to do its work. The adenosine has been building in your brain all day, and it's been whispering to you for a while now. Your core temperature has been dropping since the evening, right on schedule. Your melatonin is flowing. Your suprachiasmatic nucleus has noted the darkness and sent word to the rest of your body that it's time. The prediction machine is running, and everything is synchronized, and all those ancient systems are ready to do what they've always done. So let them. Let the slow waves come. Let your thoughts soften and blur at the edges, the way they do when you're right at that threshold between waking and sleep. That hypnagogic borderland where ideas stop making linear sense and start drifting into something looser, something more like music than language. Your breathing will slow on its own. Your muscles will release their last little holdings of tension, one by one, without you having to tell them to. And the glymphatic channels will open, and the cerebrospinal fluid will begin its quiet work, and your brain will start replaying and sorting and filing away the day. Not because you asked it to, but because that's what brains do when you finally, gently, get out of their way. This is what sleep is. Not an absence, not a shutdown. A profound, active, ancient act of restoration that you've been doing every night of your life, and that the entire history of science has only just begun to understand. And tonight, like every night, it will take care of you. All you have to do is let it.